we are looking at gene CG3961, also known as Red Queen. It's located on chromosome 3L. Functionally, this gene encodes a protein with long-chain fatty aryl-CoA's ligase activity, which means it's involved in lipid metabolism. The protein is found in the endoplasmic reticulum, and is active in membranes. There are also human orthologs, including ACSL5, ACSL1, and ACSL6. These related genes have been linked to several human diseases. For example, ACSL6 is associated with acute myeloid leukemia, ACSL5 with congenital diarrhea and other types of leukemia, and ACSL1 with obesity. From prior research in the Atkins lab, we know that CG3961 has temperature-sensitive effects. At cooler temperatures like 18 degrees Celsius, eye development is much worse. There are also sex-specific effects where males are more vulnerable than females. When the gene is knocked down, flies often show several defects. Sometimes they're headless, abnormal eye shapes, ectopic eye formation, or reduced eye size, and then many of them just don't survive. At the pathway level, CG3961 seems to be tied to the winglessness signaling, which is essential for tissue patterning. Since it disrupts the signaling in the eye, it's reasonable to hypothesize that it could also impact the wing development. And then I also attached an immunohistochemical staining from Atkins lab in 2024. This is used to show the wingless pathway at various temperatures going from the male to the female.
Hi there, my name's Caleb Shoemaker. I'll be presenting the next couple of slides about the gene CG3961 and Drosophila melanogaster. This slide, we're looking at some of the gene expression figures off of Flybase. On the left, in red, we have the cell types that it's been found to be expressed in. One of the things to take away from that figure in particular is that CG3961 is expressed across a great variety of cell types. It makes sense because it's involved in lipid metabolism. So, as you'd imagine, there's going to be a lot of different cell types that need to perform lipid metabolism. The one on the right here in blue, that one's the manually curated record of systems and tissues that the expression of this gene has been documented in. So just because one of those little squares is white doesn't mean that this gene is not expressed in that tissue. It's just that there hasn't been much work done on finding that gene expressed in that tissue. But I think the most interesting thing on this slide is the bottom figure in orange. It's a ontogenetic look at the expression of this gene, how it's expressed over time and how its expression changes over time throughout the fly's life cycle. Again, lipid metabolism, so it's expressed basically throughout the entire life cycle. But of particular interest is the fact that there is a spike in expression in the pupal stages. This is promising specifically for our research because you're going to see, of course, a lot of wing development occurring in the pupal stages. So next up, I'm going to flip in my little notes here. We'll look at some of the recent literature on this gene. We've got a few actually fairly interesting and promising studies for what we're looking at. The Poitevin study is it's looking at the role of enocytes, which are lipid metabolism cells in egg activation. One of the interesting things that they found is that knockdown of the enzymes such as CG3961 that are involved in very long chain fatty acid synthesis results in defects in egg activation, which is interesting. Of course, a lot of this stuff is interesting. Yeah, the Liu study found a link to WNT and wingless signaling. It found that WNT and wingless signaling regulates lipid homeostasis, specifically expression or activation of those results in an uptick in lipolysis and downregulation of lipogenesis and fatty acid beta oxidation. So again, another link to lipid metabolism, which is of course something that we know CG3961 is involved in. Uh, the Song paper, actually, it's looking at protein transport, specifically how something like a lipid metabolism protein like CG3961 uh, gets transported through the cell from where it's largely found on the endoplasmic reticulum to little teeny tiny intracellular lipid droplets, which is where, of course, those lipids need to be metabolized. Um, uh, it's uh, it's either during the formation of those lipid droplets that the enzymes for lipid metabolism are transported there, or afterwards by protein catalyzed formation of membrane bridges, which is pretty cool. All righty, I'm gonna hand it off for the last few slides.
Hi, my name is Sky Nugent, and I'll be presenting the last little bit of this presentation. And now that we kind of have some more background on the CG3961 gene, we can begin to look at the tools and methods we have at our disposal to guide our future experiments. So first, we can start with the tools. The first one being RNAi stalks, also called RNA interference stalks, which are genetically modified Drosophila lines that can temporarily silence or knock down genes. We have genomic or cDNA clones, also called complementary DNA clones, which they act as like a stable DNA copy of a gene, which can be inserted into vectors or DNA molecules like plasmids for studying a gene's expression and function. And lastly, a common tool to study the behavior of a gene is antibodies. But as of now, there are no known antibodies that target the red queen gene. Next are methods, the most common being the Gal4 UAS system. So, and this is pictured on the right, and it is used to study tissue-specific gene expression and knockdown. So essentially there are two lines, the driver line and the target gene line. So in the driver line, it has the Gal4 gene, which encodes for the Gal4 protein, and then the UAS target gene line, which contains our gene of interest, in this case, the CG3961. And this is upstream from the upstream activating sequence. So when these two lines are crossed, the Gal4 will bind to the UAS, and this will either express or knock down our gene of interest. And this will be activated in the tissues where Gal4 is expressed. Next, we have CRISPR-Cas9, which basically cuts and modifies DNA to which therefore alters the gene so we can better study its function. Next, we have RNA sequencing, which can be used to find like the relative abundance of RNA transcripts so we can see how active the genes are and how they're expressed under different conditions. Next, we have the FLP recombinase system, which can be used for a targeted excision of a gene. So this will make like a homozygous mutant, and this will help us to study its function and develop and how it impacts development. And lastly, we have the gene switch system, which is just a modified Gal4 UAS system, which uses the drug mifepristone to activate Gal4 to bind to the UAS. So now that we know all of this, we can start to think about some questions. So first, the CG3961 gene, it synthesizes and degrades lipids. So if this gene's not present, how could the overproduction or lack of lipids affect the wing imaginal disc, which require this transfer of lipids for development? Important signaling pathways in Drosophila like hedgehog or wingless, they require lipids for modification. So if we knock out this gene, which is required for lipid metabolism, how will this disrupt these pathways and How can this impact wing phenotype? Next, CG3961 converts the long-chain fatty acids into their active acyl-CoA form. And acyl-CoA is essential for cuticle waterproofing and wing development. So if this is not present, how will the wings be affected, and could this impact their lifespan? And lastly, how can we correlate the phenotypic outcomes of knocking out or overexpressing CG3961? And how can we associate these outcomes to the human ortholog ACSL1? And yeah, and here are the references we used for this presentation. Thank you.